Hi everyone, welcome back to the channel Dr. Hasi Explains. This is Dr. Hasi, and today's topic is BSC 1010 General Bio 1 Majors Biology and Module 4, which is all genetics related chapters, and very briefly a little review on AI literacy as well. Let's get started. Okay, so we start with the AI literacy first. Remember, so we have three Gen AI assignments that actually in one was related to the AI literacy or really responsible AI usage in education. So there are some suggestions for responsible usage. Always always align with university policies. Two, protect your personal data and only enter low risk data. Now, no identifiable human data should be entered. Three, leverage Gen AI benefits, but also mitigating the risks for improving outcomes. Four, augment education efforts, not replace them with the Gen AI. Five, beware of false information, always verify and be critical of Gen AI content. What are you verifying? Sources, citations, authorships, uh, pretty much evaluate everything, verify, edit, revise, make sure, and also number six, be transparent about usage of Gen AI, as um, uh, including using chatbots as a tutor, 24/7 tutor, using explain features or quiz me features or some other features. So for output, you still need the critical eye of the whether information is correct or not. And again, you need to use good descriptive prompts like advanced prompt, one of the assignment three asks you to do specifically to test simple prompt versus advanced prompts. That's Gen AI responsible usage in education. So please use it responsibly. Now we are going to start with gene expression, uh, chapter 17. Again, this is module 4, which includes chapters 13, 14, 15, and 17. Okay, so we will start with the 17, which is gene expression. So gene expression is about two major processes, which are um, transcription. We talk about eukaryotic gene expression and translation. So transcription usually starts with we start with the nucleus. You see this bubble here. You should study this, chapter 17. So there is gene here, which is part of the DNA. There are parts of this gene structure, like upstream parts, like promoters, like core promoters, transcription factors, and RNA polymerase 2, like you see here, which is the enzyme. It starts the transcription of DNA, eventually produces this single-stranded mRNA or messenger RNA. Then mRNA needs to be, after transcription, spliced the introns, and then eventually get shorter as mature mRNA, leave the nucleus, go find a ribosome, and mature mRNA gets into the ribosome, especially the coding strand, template strand, and then antisense or non-coding strands synthesized to mRNA here, and then mRNA goes and um, into the ribosome and then synthesized into or translated into protein. You can see here this blue amino acid group. Um, so this is basically chapter 17, and you should read more from your textbook and take notes and using this infographic of chapter 17. Chapter 15 is chromosomal basis, which is about chromosomes and a lot of non-Mendelian genetics for inheritance, including multiple alleles, um, and also polygenic inheritance. And specifically, it's about chromosomes. So, and the fruit flies or Drosophila is the main model system here in chapter 15. As you see here in one map, you can see specific genes of the fruit flies and their location in specific locus. This is map, and this any two genes are on a map in the same on the same chromosome, they are considered linked. And if two genes are linked, they do not necessarily follow Mendelian's rule because they are linked. And like a number of other genetic situations don't follow Mendelian rules, there are exceptions to Mendel's laws, including codominance, incomplete dominance, again, gene linkage, which can give you unexpected ratios, non-nuclear genes, like genes from chloroplast, and some other environmental effects. So 15 is very, very important chapter. You have to understand also 14 to really understand 15. There is also this formula, recombination frequency equals number of recombinants divided by total number of offsprings times 100. And that can be converted to a physical distance on a chromosome, and that can help us to actually find the locations of specific genes on a specific chromosome and map those genes. Chapter 14 is the Mendel genetics because it's a basic genetics starting with Mendel's two flower colors, parents, and then crossing them and looking at F1 and selfing the F1, look at the F2, and then finding 3-to-1, 3 dominant, 1 recessive ratio, which tells us this is one gene. Chapter 14, students should actually do some vocabulary, like a list of the vocabulary and study individual vocabulary, including alleles, genotype, phenotype, homozygous, heterozygous, dominant, recessive, X-linked recessive, and then specific Mendel laws of dominance, segregation, and independent assortment. Mendel worked on genes that their alleles had some dominance. Some alleles were dominant, some are recessive. Recessive doesn't mean it's any less than dominant. It just means there are 2 copies needed in order to recessive show. Blue eye color is a good example, which is a little more than 5% of the world population have blue eye color, which was originally a mutant 10,000 years ago. And today it's a recessive trait, a good example. X-linked, in plants, we don't see X chromosome, but X-linked genes are very important in chapter 15. Because males have only one X chromosome, they will more likely express a recessive trait if it is located on X chromosome. This is about chapter 15, but related to 14 too, but it's a non-Mendelian situation. And then the other chapter was chapter 13. Chapter 13 was about life cycles and meiosis. A life cycle might look like this. It's very simplified. 2 processes, meiosis, fertilization, 2 big parts of life cycle, sporophyte, gametophyte. Sporophyte goes through meiosis and produces 1N gametophyte, and 2 gametophyte, micro, mega, actually go and fertilization produce diploid sporophyte. So every life cycle has these 4 parts, but have other parts too. We should study, pay attention to what a plant life cycle versus animal versus a fungi life cycle. So that's, and also remember 10 steps of meiosis in how it goes: interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis again in both cases, and then how half the chromosome number, the 4 daughter cells produced here, they are actually unique. They are not identical themselves. Also, they are not identical to the parents. So that's chapter 13. Keep studying. We will do a big review also in class after we finish this last chapter. So keep studying chapter 17 right now, which is gene expression. Pay extra attention to Mendelian genetics 14, and chromosomal theory 15. To just to recap, this is module 4 of Majors Biology BSC 1010 and includes all genetic chapters. So please feel free to comment, like, and subscribe. And see you next time, and happy studying.